stratum basale at the base. Going up, it becomes and then spinosum, and then corneum, right? That is for a simple skin that you have. But in most vertebrates, look at most of their epidermal part have various modifications. There could be mucous glandular glands, there will be germinative glands, and even in the dermal part, these are your epidermal layer. And your dermal layer will be what? Composed of the papillary type, the papillary dermis. Okay, this is for humans, okay? And your reticular dermis. And how about in most vertebrates, na ay additional term, which is found in the dermal part, the stratum laxum and the stratum compactum. These are notable layers of perhaps all, or generally speaking, to all vertebrates that makes vertebrate scale very dry, very sturdy, very keratinized because of this notable layer, okay? Especially in this part. What can you say about the layers here? This is composed of mucous and glandular gland. These are notably slimy type because you notice that these are the glands that usually produce mucus and certain degree of secretion, I mean, which makes the skin more or less mucus or what do you call that, slimy to the touch. And some notable cell modification, especially in the rainbow lizard. From the name itself, what do you think the rainbow lizard will appear like? They have notable cells that gives color. We call them the pores, the melanophoric types of cells, especially if it gives of dark, yellow, and red types. And the iridophore is more or less it gives a bluish tinge or purplish tinge. And these are the cells involved as to the diversity of the colors of some of the notable, let's say, lizards. Okay, we'll not use rainbow color. Have you seen a chameleon? Which basically a reptile, right? A type of lizard. And you can actually expect them to appear in various colors because they are king of camouflage. These are the types of notable cells, which you don't have, by the way. Okay? Can you change color? Yes? Kasi may buong lang ko ni Carla, palit na kakaputi ni blend kas buong. Okay? So, these are notable modifications in the typical vertebrate integuments. Okay? In this particular layer, this is the development of your papillary dermis, characterized by, first, we have the mesenchymal papilla at the embryonic level. And then, this is basically leads to the formation of what you think is the notable structure here. This is dentin formation. When you say dentin, this is another known, lesser known integument. Basically, these are your teeth. And you'll notice that across vertebrates, teeth varies. But at this point, you should have an idea what are teeth. Okay, so many mga teeth na they are, how do you call this part? This part and the back part. Can you name some teeth? Molars, premolars, canine, and incisors. Okay? But you'll notice that not all of these teeth are shared across all vertebrates. You don't expect teeth of carnivorous animals to have the same to that of an omnivorous one. Okay? Because most omnivores and herbivores will have mostly dominated by molars rather than canines. And you'll notice that carnivores would expect more of canines rather than molars, of course, because it is something to do with their predatory habit. Okay? So those are the kinds of modification that you should know. Okay? So what else? A notable skin of, let's say, a shark. Okay? And this is what I am emphasizing. It has a horn. Okay? So this is a type of scale that is notable in shark. Though our shark model there is not a very good candidate to feel this type of scale because it has been worn off over time due to constant touching. But if you happen to touch an actual shark, you can actually feel its skin. And it is true even to some members of the skates and the stingray family. Medyo rough silang skin. Okay? Because of their placoid type of scales. Alright? So, you'll notice that in this case, eye part of their skin has a dentin-like formation. So it has structure similar to that of a teeth. So, sila enamyloid type, meaning they are studded with teeth-like projections on their skin. Okay? But somehow, in higher vertebrates, the type of phenotype has been concentrated to the jaw part already. But in sharks, you'll notice that it has, what do you call this? It covers its body. Okay? And then what else? The skin, the bony scale of the teleus. What are teleus again? And then the actinopterygian. These are your members of the early fishes, right? These are your lobe fin fishes. Okay? So what you'll notice is that most of these fishes have hardened skin or rough skin. Okay? So in our lab today, just feel free to touch the surfaces of the models of the representative vertebrate. Okay? So what else? You have your cycloid and your tenoid scale. What are these? These are the scales of fishes. Okay? Alright, so what can you say about this one? The identity of the cycloid, under the microscope, we'll have a microscopic viewing of the types of the scales. You can actually see that a cycloid, coming from the word cycle, this is actually a scale ring. Okay? The more the fish grow, the more it creates ring in order to create a notable scale. This would actually help you age the fish. It is cycle because you can actually see concentric ring that starts at the focus and then emerging outward towards the anterior margin. Where in comparison to the tenoid scale, these are the rough edge scale. Okay? These are the type of leptoid family. Okay, these two are called as the leptoid scale. These are the type of scale that has no notable protrusion. They're just flattened. Okay? They can just base them based on the edges. So the ones that are more edgy are the tenoid scale, which are usually attached to the chromatophoric end or the posterior margin, which contains the tenine. It's actually the one that is attached to the scale. Most visibly so in most fishes here. Okay? Especially the posterior margin. So this is the embedded part, and this is now the exposed part. And you can actually see the edges here of the fish later on in the microscopic viewing. Okay? Alright, so what are these? These are your notable vertebrate skin, especially the members of the amphibian and the reptilian side. Notable for having chromatophoric, laden with chromatophoric structures, blood vessels, and all. Noticeable that instead of the papillary dermis and the reticular dermis, it is replaced by a stratum laxum and a stratum compactum. Why is it called compactum? Because instead, have you seen the have you seen a skin under the microscope? For some part, okay, we will be able to see a skin. I want you to compare the frog skin and the human skin in terms of how compact the fibers in this layer. And you'll notice that in frogs it is more compact. Notable compression of the fibers creating a compact layer. And then a laxum, what do you call this one? Laxum, from the word lax, meaning it's more, the fibers are more loose. Okay? This is completely different by the way the papillary and the reticular arrangement in the human skin, because the papillary dermis in human skin is more compact on the papillary side and it is lightly loose on the reticular side. So bali siya sa reptiles. You'll notice that most of the loose part are on the surface, giving some of the skin of reptiles a bit soft. You know, it's a bit edgy, a bit hardened, but at some point you can actually feel its slightly slimy touch. Okay? You'll notice that I do hope that we'll still be able to invite an invertebrate group which brings in pythons and snakes, and you have the time. You can actually touch a tarantula, so on and so forth. We're planning that in order for you to have an experience. How about in safari, are you able to touch a snake? Only birds, right? I think there's one group. Well, I think it was Api who got the chance to touch the parrot. Alright, so yeah. But more or less, manta ito makatouch ng skin because of the inherent danger of touching reptiles, hindi kaya nato siya i-allow in the lab. No, lagi sa magdako niya buhaya, binti, naroon na gito. Nalang siya the imaginative part. Okay, but the feather is still okay. Okay, kay especially si Carlisle, magtatarik man to si Carlisle. Kato yung example, nagpuput siya manok iksasabong, for the example, right? Yes. So, yeah. So take a look at the down feather, the pellucose, and the contour feather in the lab. We have a bird representative. Okay, and this is your typical human skin. And look at how compact the papillary side to that of the reticular side. Well, I can emphasize it. Okay, so notable on the layering of the skin. Okay, and so on and so forth, and some of the notable modified structures. Are you aware of this? The sweat gland, the eccrine gland, okay, the root hairs and bone. You'll be able to see that in the lab. Okay. Wala ni answer, did any group answer a nipple or an areola? Would you consider the nipple or an areola as a integument? Yes. Yes, very good. Because the areola here, or the nipple, is actually a modified epidermal structure, no? But somehow attached to a developing gland, which later on becomes the mammary gland, which produces milk. These are needed for suckling, especially in mammals. Okay, mammals are known to be good mothers. You are a mammal. Okay, you were cared for by a mom. So, yes, your breast, or particularly the nipple areolar complex. Where's the nipple there? The protruding part, and the areolar complex are the pigmented outer rim, constitute still an integument, notably found in mammals, and we call them teeth. Okay, especially teeth. Dili po teeth ng nipple na. But nevertheless, you use your teeth to suckle your teeth. Teeth is to teeth. Okay? Alright. Alright. So, what are the notable modifications here? Okay. Most of the ancient fishes are notable for having dermal armor. But in most cartilaginous fishes, the Collectarius, presence of the placoid scale, which later on tends to become more soft, smooth on the surface, and then later on becomes the, what group is after the lungfishes in the overall vertebrate clade? What's after this one? It evolved into? So after yung mga lungfishes, katong mga tumilakan with notable limbs. These are your salamander, right? Yeah. Right? The evolution of the true limbs after the evolution of the lungs and this coelacanths, okay? The notable lobe fin fish, which later on becomes the limbs of tetrapods. Okay. Alright. So, in your lab today, I want you to explore. There are demo slides at the back of the room in order for you to explore the notable dermal structures, may it be a feather, or a scale, and then later on you'll get again the preserved models and try to examine the surface scale. And I want you to use the exercise 4 in order to understand the important terms there. Have you had a copy? We have a copy of your exercise 4. Okay? So you will see in the lab the frog skin. Okay, I don't know if you have, I'll check if you still have the armadillo. Okay. You know what's an armadillo, right? Yes. Okay. Human skin and the dermal scales of some notable fishes. The turtle exoskeleton. The birds of feathers. And then the exoskeleton of mammals. Okay? So that's it. Alright, so let's have a 10-minute break before going to the lab. Okay? So our lecture part is